The overall goal of this procedure is to introduce undergraduate science students to microarray technology by examining the expression differences in yeast undergoing oxidative stress. This is accomplished by first isolating total RNA from yeast cultures treated with hydrogen peroxide and the untreated controls. The second step of the procedure is to generate and purify cDNA from these RNA samples. The cDNA is then used to prepare biotin-labeled cRNA via in vitro transcription. The final step of the procedure is fragmentation of the biotin-labeled cRNAs for hybridization to the Affymetrix yeast gene chips. Ultimately, results can be obtained that show the expression differences in the two yeast samples, and through bioinformatics demonstrate the power of microarray analysis to undergraduate students.
The Vermont Genetics Network outreach team first had the idea for this method when they were charged with delivering microarray technology to science students throughout the state of Vermont in order to enhance their science education. To date, this method has been delivered to multiple sites at 8 baccalaureate colleges throughout the state.
So the main advantage of using microarray of a teaching module is it gives us the ability to teach general molecular biology techniques that are used every day throughout the lab to a group of individuals. And we also get to use this to teach to students and teachers alike. In our groups, we actually have students and professors working together on the same project. They learn certain techniques that take a lot of finesse, like handling RNA, which is not an easy task. They learn how to precipitate DNA using visualization reagents. They actually get to use the techniques that they're going to encounter in graduate studies or in everyday molecular biology labs.
Though this method is suitable for gaining insight into oxidative stress in yeast, it's certainly